Now I'm very sure that you all have studied about what primary metabolites are. So I explain you once again that primary metabolites are the chemicals, or they are the organic molecules which are synthesized by the normal metabolic processes of the plant. Say for example photosynthesis, respiration, say for example pyruvate, erythrophosphate phosphate, okay, citric acid. So these are the metabolites which are required in the normal metabolic process of the plant, okay, and they are found in all living systems. They are common to all plants and they are found in all living systems, and these primary metabolites, they are essential for the growth and survival of the plant. Okay, so these primary metabolites are essential for the growth and survival of the plant. Okay, as I said, the example include the metabolic substances from the process such as respiration, photosynthesis. So these examples can be grouped into amino acids, nucleotides, sugars, acyl lipids, okay, which are required all the time by the plants and they are common in the plants. Okay, but what are secondary metabolites? So I here write one difference, that secondary metabolites are not required all the time. Okay, and they are produced under specific conditions. They are produced under stress conditions, and they are not produced all the time, and they do not play any role in growth. Okay, they do not play any role in growth, and they, what do they do then? They help in defense responses. Okay, so what we studied, that there are two kinds of organic molecule produced by the plant. One is primary, this secondary. Primary metabolites, they are the intermediates which are found in all living system like amino acid, nucleotides, sugars, and these primary metabolites helps in the growth and survival of the organism, and secondary metabolites are produced only in the stress conditions to protect the plants against certain stress responses. Okay, they do not help in any growth. Okay, so see, they have a restricted distribution. Secondary metabolites have the restricted distribution in the plant kingdom. Now what do you mean by restricted distribution? That means that particular plant will produce a particular type of secondary metabolite. Okay, there are different kind of secondary metabolites, and a particular secondary metabolite is restricted to one group of plant only. Okay, different plants produces different kind of secondary metabolites to combat with the stress. Okay, so they have restricted distribution in the plant kingdom, that is particular secondary metabolite are often found in one plant species or related group of species, but primary metabolites are found throughout the plant kingdom. Okay, so primary metabolites are common to every plant like amino acids, sugars. Okay, but secondary metabolite, we will see the examples later, they are restricted to particular group of plant. Like group A produces one kind of secondary metabolite and group B produces another kind of secondary metabolite which is not produced by group A. Correct? Now I already told that they are not required for the growth and they are only produced under stress or the adverse conditions. They help the plant to defend against herbivores and pathogens. They serve as attractant for pollinators. Now they have specific properties, okay, which attract the pollinators. Like some secondary metabolites give the color to the plant. Okay, we will see which metabolites. So some gives color to the plant. So in that way they serve as attractant for pollinators, and when they are serving as a purpose for pollinators, they also help in seed dispersing. Okay, and they act as an agent for plant competition. Okay, some plant will synthesize a kind of secondary metabolite and spread it in the whole area, and will not allow the plant living nearby to grow. Okay, so some plant will release some kind of secondary metabolite which will act in the nearby areas of that plant and will not allow another kind of plants to grow in that area. So this in that way they act as an agent of plant competition. Okay, so see here we see that there are different kinds of metabolites. Now one is terpene, second is phenolic compound, third is nitrogen containing compound. Okay, so secondary metabolites are divided into three major groups: terpenes, phenolics, and nitrogen containing compounds. We have to learn the biosynthetic pathway of these three major groups. What is the name of the pathways by which these groups are synthesized? Okay, so here I have summarized. See, so this terpenes, I mark it number one, first kind of secondary metabolite. Okay, these terpenes are synthesized by MEP and MVA pathway, if I make it short. So mevalonic acid pathway and MEP pathway. Terpenes are synthesized by mevalonic acid pathway and MEP pathway. About the precursors we will see later when we will study about each secondary metabolite in detail. Then we will go from the beginning. Now we are just trying to understand the different names, different biosynthetic pathways by which each of the secondary metabolite is synthesized. So terpenes are synthesized by mevalonic acid pathway and MEP pathway. Phenolic compounds are synthesized by two pathways. One is malonic, so I name it number 2, mark it number 2. Phenolics are synthesized by malonic and shikimic. See, the arrow is going here. Arrow is going here. So phenolic compounds are synthesized by malonic and shikimic, and nitrogen containing secondary products are synthesized from aromatic acids. Okay, so this is the biosynthetic pathway of secondary metabolites. Okay, I have just told the names. We will go in detail in few slides when we will study about each secondary metabolite in detail. So see here I give you some example of primary carbon metabolism or primary metabolites also. So example of primary metabolite include here pyruvate, 3-phosphoglycerate, acetyl CoA, then this aliphatic amino acid, erythrose 4-phosphate. Now these are common to every plants. Okay, since they are common to every plants and they are common throughout the plant kingdom, they are said to be as primary metabolites. Okay, so the blue colored ones are secondary metabolites, and the pink colored ones are primary metabolites. Okay, now if we summarize the three biosynthetic pathway of three different types of secondary metabolites. So the first type of secondary metabolite is terpene. So there are three groups. Okay, so here I write the name and here the pathways by which each are synthesized. Now this is very important to know because question comes directly straightforward from this topic. So terpenes are synthesized by mevalonic acid pathway and MEP pathway. Now MEP pathway is also known as methyl erythritol pathway. Methyl erythritol pathway. Okay, so in short it is known as MEP pathway. Now second class, phenols, phenolic compounds or phenolics. Okay, now this phenolics are synthesized by again two pathways. Number one is shikimic acid pathway. Please remember all the names to solve the question. Shikimic acid pathway, and another is malonic acid pathway. Okay, among the two, this is the most important and this is found in higher plants. Okay, number third type is nitrogen containing compound. Nitrogen containing secondary metabolites. Okay, and they are synthesized from amino acids. Okay, so please remember terpene from mevalonic and MEP pathway, phenolics from shikimic and malonic acid pathway, and nitrogen containing secondary metabolites, say for example alkaloid. Okay, they are synthesized from amino acid. Okay, so starting with the first secondary metabolite named as terpene. So what are terpenes? Terpenes are the largest class of secondary metabolites. Why they are called the largest? Because these small unit of terpene known as isoprenoid unit, okay, they can form large number of compounds. Okay, so I tell you that these terpenes are formed by a unit known as isoprene unit. Okay, these isoprene units are 5 carbon compounds, and these isoprene units, which are 5 carbon compounds, they combine with one another, like one isoprenoid combined with another one isoprene combines with another isoprene to form 10 carbon compound, and 10 will combine with another isoprene unit to form 15 carbon compound. In that way they can form large number of terpenes. Okay, by different combination of isoprene units they can form large number of terpenes. So terpenes are the largest class of secondary metabolites. They are insoluble in water. Now terpenes and terpenoids have a difference. Terpenes are the hydrocarbons. Okay, hydrocarbons means hydrogen directly bonded with carbon. And terpenoids are oxygenated hydrocarbons. Okay, oxygenated hydrocarbons means they have oxygen with the hydrocarbon. Okay, so terpenoids have oxygen with the hydrocarbons. Now these terpenes are also known as isoprene units. Okay, what is isoprene unit? One isoprene unit contains 5 carbons. Okay, and one 5 carbon isoprene is known as hemiterpene. Okay, one 5 carbon isoprene is known as hemiterpene. Now they are biosynthesized from acetyl CoA or glycolytic intermediate. So the precursor of IPP, isopentenyl pyrophosphate, is acetyl CoA or glycolytic intermediates. Okay. So what we discussed, that terpenes are the largest class, they are insoluble. Terpenes are only hydrocarbons and terpenoids are oxygenated hydrocarbons. Now these terpenes are also said to be as isoprene units, okay, because when terpenes are kept at high temperature, when large terpenes are kept at high temperature, they break and form isoprene units. I'll show how. And one isoprene unit is known as hemiterpene. Okay, 5 carbon isoprene unit is known as hemiterpene, and the precursor of terpene is acetyl CoA or glycolytic intermediates. Let's see how. So first we will do the classification of terpene. So see, I told that terpenes are formed by the fusion of 5 carbon isoprene units. So all isoprene units are 5 carbon compounds. Okay, so they are formed, many isoprene units are fused together to form large terpenes. Okay, one isoprene unit will form hemiterpene. Several isoterpene units will fuse together to form large terpenes. Say for example, isoprene units means what I said, IPP. Okay, say for example 10 carbon terpenes. 10 carbon, how it will be formed? 1 5 carbon plus another 5 carbon will form 10 carbon terpenes. Okay, and this 10 carbon terpenes are known as monoterpenes. Okay, this 10 carbon will combine with another 5 carbon. Okay, so I'll say that 10 carbon plus another 5 carbon will form what? 15 carbon. Now this 15 carbon are known as sesquiterpenes. In the third case, 4 C5 units, or in the case I can say that 2 times when monoterpenes combine, or I can say 10C plus 10C, or 2 times monoterpenes when it combines, it forms diterpenes. Similarly, if 2 sesquiterpene, one sesquiterpene is of 15 carbon. So if 2 sesquiterpenes combine, it forms triterpene. 2 sesquiterpene means what? So if I write triterpene, it is of C15 plus C15. One C15 is sesquiterpene and 2 sesquiterpene will form triterpene, which is C30. This is how tetraterpene will be formed. 2 diterpenes will combine to form tetraterpenes. And polyterpenoids are when there are more than 8 isoprene units. Okay, when there are more than 8 isoprene units, it is polyterpene. Okay, in tetraterpenes, how many isoprene units are there? In tetraterpenes, there are 8 isoprene units. Okay, like 20 plus 20. So this is 4 into 5 carbon, and this is 4 into 5 carbon. 5 carbon means isoprene units. So total tetraterpene has 8 IPP. Okay, 5 carbon means IPP. So total tetraterpene has 8 IPP. So up to 8 it will be tetraterpene, but when more than 8 it will be named to be as polyterpenoids. Okay. Now how these terpenes are synthesized? Okay, what I told, that the precursor is what? is IPP, which is isopentenyl pyrophosphate or isopentenyl diphosphate. They are the same thing. But how this IPP is synthesized? So IPP is synthesized from where? IPP is synthesized from acetyl CoA or another glycolytic intermediates. Okay, IPP comes from where? IPP comes from acetyl CoA or glycolytic intermediates. Now see the picture. So see, when three molecules of acetyl CoA combine, it forms mevalonic acid. Okay, so I told two pathways are there. One pathway is mevalonic acid pathway, this one. Another pathway is MEP pathway, which is methyl erythritol pathway. So two pathways are responsible for terpene biosynthesis. Here also I have named two different pathways. Okay, MEP and mevalonate or mevalonic. Okay, so in the first pathway, what happens? 3-acetyl CoA will combine and form mevalonic acid. This mevalonic acid will form IPP. In methyl erythritol phosphate pathway, what happens? Glyceraldehyde triphosphate and pyruvate, which are the glycolytic intermediates. Okay, so they are what? They are glycolytic intermediates. They combine to form what? Methyl erythritol phosphate, which will form DMAPP. Now what is DMAPP? DMAPP is an isomer of IPP. Okay, so DMAPP is an isomer of IPP. So what we studied, that acetyl coenzyme A, it acts as a precursor. Where from acetyl CoA came? Acetyl CoA came from TCA cycle. So acetyl CoA, which acts as a precursor. Three molecules of acetyl CoA are joined stepwise to form mevalonic acid. Now what are the steps? There are several steps in between. What are the steps here? So the first step here is pyrophosphorylation. Second step is decarboxylation. I'm very sorry. Acetyl CoA3 molecules of acetyl CoA will combine to form mevalonic acid, so which is a 6-carbon compound. Now this is forming IPP, which is a 5-carbon compound. IPP is C5. So how from 6-carbon 5-carbon compound was formed? So here there are 3 steps. Okay, I'm writing it here. So here there are 3 steps involved in the conversion of mevalonic to IPP. So what are the few steps? One is pyrophosphorylation. Second is, since 6-carbon is forming 5-carbon, so it will be decarboxylation. And the third is dehydration. One molecule of water is removed. Okay, so these 3 processes are working here. Okay, which is ultimately forming what? Which is ultimately forming what? This is forming IPP. Okay, please remember the sequence, very important. Please remember the sequence. They occur in this sequence only. pyrophosphorylation, decarboxylation and dehydration. Okay, so this up to here is the path. After IPP the pathway is same, so we have to first understand the precursors. Okay, so this pathway is clear, the mevalonate pathway is clear. Now coming to methyl erythritol pathway. So I would like to mention that the first pathway, which is mevalonate pathway, this occurs in the cytoplasm. And the methyl erythritol pathway, this occurs in chloroplast. Okay. Now, in methyl erythritol pathway, what we studied, this can be formed from the intermediates of the glycolysis or photosynthetic carbon reduction cycle. Okay, they occur in chloroplast and some other plastids. Now, glyceraldehyde and 2 carbon atoms derived from pyruvate. Okay, they combine to generate an intermediate which is MEP. This intermediate will be forming DMAPP, which is dimethyl allyl diphosphate. Okay, here the enzymes required are DOX-P synthase and DOX-P reductase. Okay, since DOX-P enzymes are required, this pathway is also known to be as DOX-P pathway or DOX-P pathway. Okay. Now this methyl erythritol pathway is also known to be as non-mevalonate pathway. This is mevalonate pathway, this is non-mevalonate pathway. Okay, or mevalonate independent pathway. So clear? So after IPP and dimethyl allyl diphosphate is formed, this 2,5 carbon compound. Okay, so IPP from this pathway, okay, this IPP will isomerize to form DMAPP. So always one molecule of IPP and one molecule of DMAPP will combine. Okay, so this DMAPP which is synthesized here, one molecule of DMAPP, it will be isomerized to form IPP. Okay, so IPP and DMAPP will combine to form a 10 carbon compound. The name of this 10 carbon compound is geranyl diphosphate, GPP. And this 10 carbon compound already discussed, it's a monoterpene. Now this 10 carbon will combine with another molecule of IPP. Okay, 10 will combine with one molecule of IPP to form 15 carbon compound known as farnesyl diphosphate, FPP. Now this 15 carbon is a sesquiterpene, which is C15, and two 15 carbon compound will form triterpene. Okay, C15 plus C15 will form C30, which is a triterpene. Now this C15 will combine with another IPP. Now IPP is 5 carbon, you all know. Farnesyl will combine with another IPP to form 15 plus 5, 20 carbon compound. The name of this 20 carbon compound is geranyl diphosphate. And this geranyl diphosphate is a diterpene. And two diterpenes will form tetraterpene, which is C40. Okay, so tetraterpene is C40, triterpene is C30, monoterpene is C10, and diterpene is C20. Okay, now this geranyl diphosphate, which will form, again when more IPP are combined here, it will form polyterpenoids. Okay. So what we discussed, that there are two pathways, mevalonic acid pathway and methyl erythritol pathway. In short, it is known as MEP pathway or DOXP pathway. Okay. Now this mevalonic acid pathway occurs in cytosol, I discussed. It occurs in cytosol. First is in cytosol, second is in chloroplast or plastid. Here acetyl coenzyme is the precursor. So 3 acetyl molecules, which is 6 carbon. Okay. 3 into 2. 6 carbon will form what? Mevalonic acid. This mevalonic acid will undergo pyrophosphorylation, decarboxylation and dehydration to yield IPP. Now IPP is a building block of all terpenes. Please remember this. So again, what we discussed, that one IPP plus DMAPP. So IPP will isomerize to form DMAPP. Okay, to form what? 10 carbon compound, which is GPP. Okay, this GPP is geranyl diphosphate. Now geranyl is a precursor of all monoterpenes. Now this GPP, which is 10 carbon, it will combine with another IPP to form what? 15 carbon compound. The name will be farnesyl pyrophosphate, which is 15 carbon compound. Okay, and it is a precursor of all sesquiterpene. Now 15 carbon compound will combine with another 5 carbon compound IPP to form 15 plus 5, 20. The name of the 20 carbon compound is GGPP, double G. Geranyl pyrophosphate. In mono it's single G. In diterpene is double G, which is a 20 carbon compound and is a precursor of all diterpenes. Okay. After that, several FPP. Finally, FPP can dimerize. One FPP plus another FPP. FPP was 15. 15 will combine to form 30 carbon compound. 30 carbon compound is known as triterpene. And this diterpene, which is GGPP, they can dimerize. So GGPP was 20 and 20 will combine to form tetraterpene, which is C40. Okay. Now in MEP pathway, already told that this occurs in chloroplast. Okay. And here there is a reduction cycle, which is DOXP reductase. Okay. It occurs in chloroplast and plastids. So glyceraldehyde 3-phosphate and 2 carbons from pyruvate, they generate an intermediate. Intermediate was what? Intermediate was MEP, methyl erythritol phosphate. Okay, which is converted to the isomer of IPP, which is dimethyl allyl pyrophosphate. Here the enzyme required is DOXP synthase and DOXP reductase. So the name was also DOXP pathway, also known as non-mevalonate or non-mevalonic acid pathway. So what we studied here, that one is hemiterpene, which is a 5 carbon isoprene unit. Second, what we studied? We studied monoterpene, which is a 10 carbon compound. 2 IPP. Third is sesquiterpene, which is a 15 carbon compound. It has 3 IPP. Okay. Fourth is diterpene, which is 20 carbon compound, which has 4 IPP. 5 is triterpene. Triterpene will have 6 isoprene unit. 2 sesquiterpene when combined together, it forms triterpene. So 2 sesquiterpene. So 2 into sesquiterpene can form triterpene. Next, tetraterpene is 40 carbon compound, which is 8 into IPP. Okay, so 2 diterpenes can combine to form tetraterpene. Okay, next is, so if I say N is equals to IPP, if I say N is equals to IPP, so this is 8 into N, 6 into N, 4 into N. So last is polyterpene or polyterpenoids. Okay, they contain N greater than 8. That means more than 8 IPP will form polyterpenes. Okay, so what we studied? We studied 2 pathways. One pathway was mevalonic acid pathway and second pathway was MEP pathway. Okay, in mevalonic acid pathway, precursor was acetyl CoA, and in MEP pathway, the precursor was glyceraldehyde 3-phosphate and pyruvate. Clear? Now coming to some important terpenes which fall under these class. So I'll highlight only the important ones. Okay, so monoterpenes, for example, camphor. For example, the spelling is wrong here. For example, camphor. Okay, limonene, menthol, they are under monoterpenes. Okay, this camphor and limonene, they have medicinal value. Okay, one more, pyrethroids. So pyrethroids also fall under monoterpenes and they have insecticidal activity. Pyrethroids are synthesized in the leaves and flowers of chrysanthemum. Okay, so these monoterpenes example include camphor, limonene, pyrethroids. Okay, next include diterpene. If I mark gibberellin, which we have studied already, gibberellin is a diterpene, 20 carbon. Okay, 20 carbon compounds. And another example of diterpene is Taxol. I am marking only the important ones. Okay, Taxol. This Taxol have anti-cancer activity. Okay. Third is sesquiterpene. So abscisic acid is a sesquiterpene. It is a 15 carbon compound. Okay. And next is triterpene. So example include sterols, saponins, sterols, racinosteroids we have done already. Now these sterols are triterpenes and they are essential component of cell membrane. which they stabilize by interacting with phospholipid. Okay, they are the essential components of cell membrane and they stabilize it by interacting with phospholipids. Another example of triterpene is cardenolide. I'll describe what cardenolides are. Cardenolides, Cardenolides. Okay. Now coming to polyterpene, these tetraterpene are carotenoids, okay, which gives color to the plant. Okay, there are terpenes which give color to the plants. Now last, which is not included here, are polyterpenes, where N is more than 8. So the example include dolicol, chlorophyll, okay, rubber. Okay, so dolicol are what? Dolicols, they function as a carrier of sugar in cell wall and glycoprotein. If you have studied the membrane chapter, okay, they function as a carrier for sugar in cell wall. Now phytol chain of chlorophyll is a polyterpene. Okay, this phytol chain help anchor certain molecules in membranes. Okay. So what we studied, for the most common one for monoterpene is pyrethroid, okay, which has the insecticidal property, and this pyrethroid have very low persistence in the environment. They are of negligible toxicity to mammals. Okay, that is why they can be the ingredient of many insect repellent. Limonene and camphor, which have medicinal value. Okay, diterpene, the main example was Taxol. Okay, vitamin A1, gibberellin. Again, what we studied, sesquiterpene. Sesquiterpene, the main hormone ABA, which we studied already, this is C15 compound. Okay, so next was triterpene, tetraterpenes, which are what? Carotenoids, mainly beta carotene. Triterpenes are what? Triterpenes include sterols. Okay, brasinosterolide is again a hormone which is a sterol. Now polyterpenes, it includes what? Rubber, chlorophyll, okay, dolicol, which are the sugar carriers. So these you have to remember by heart. Okay. So one question, which of the following acts as a branch point for the biosynthesis of sesquiterpene and triterpene? Triterpene is C30, sesqui is C15. Okay, so which one is the branch point? So we discussed geranyl pyrophosphate is a monoterpene, which is C10. Isopentenyl is C5. Here this is not related. So farnesyl is C15. Okay, so C15 will form sesquiterpene also. C15 will form triterpene also. So this is a branch point of biosynthesis for sesquiterpene and triterpene. Next question, in terpene biosynthesis, 3-acetyl CoA are joined together stepwise to form mevalonic acid. Which one of the following 3 steps is required by mevalonic acid to form IPP? So the 3 steps were discussed. It is phosphorylation, decarboxylation and dehydration. Now some important functional roles of terpenes. They are toxin and feeding deterrents. Okay, they do not allow the animals to feed on that plants which produces the terpenes. For example, some plants produces terpenes named as pyrethroids. These pyrethroids are monoterpenes. Where are they produced? They are produced by the chrysanthemum leaves and flowers. The leaves of and flowers of chrysanthemum produces monoterpenes known as pyrethroids. These pyrethroids are extracted. These pyrethroids are helpful for the chrysanthemum plant also, and they are extracted from chrysanthemum plants also to act as an insecticidal ingredient. Okay, so they have the insecticidal activity. So both natural and synthetic pyrethroids, they are the popular ingredients in commercial insecticides. Why they are the popular ingredients? Because they have their low persistence in the environment. Okay, and their toxicity is negligible to mammals. Pyrethroids toxicity is negligible to mammals and they have low persistence in the environment. But they are insect repellent. Okay, some monoterpenes accumulate in the resin duct. Where are these resin ducts found? These resin ducts are found in needles, twigs and trunk. Okay, and these monoterpenes are toxic to numerous insects. Which insects? Bark beetles. Now these bark beetles are the most common pest of the conifers all around the world. Okay, so conifer plants produces monoterpenes to combat with the bark beetles. Next, some compounds produces volatile monoterpenes and sesquiterpenes, and these volatile terpenes are known as essential oils. Very important. Now, what are essential oils? These essential oils do not allow the pathogen to attack the plant. Okay, say for example, what are essential oils like peppermint plant, lemon, basil and sage. These are the plants which produce essential oils in response to the pest. Okay, these essential oils are volatile terpenes, and volatile terpenes can be monoterpene or sesquiterpene. Okay, so the chief monoterpene constituent of peppermint oil is menthol, and that of lemon oil is limonene. These essential oils have insect repellent properties. Okay, and they are found in the glandular hair that project out of epidermis. If this is the epidermal layer in the microscopic view, so there is a glandular hair that project out from the epidermis. Okay, and these essential oils are found in the glandular hair. Okay, next, there are some non-volatile terpenes also, which includes limonoids. So limonene was volatile. Limonene was volatile and limonoids are non-volatile. This limonene was monoterpene. Limonene was monoterpene. And limonoids here are triterpenes. Limonoids are the bitter substances in citrus fruits. Okay, one kind of limonoid is azadirachtin, which is a kind of limonoid produced in neem plants, also. And because of this limonoid in neem plants, they have commercial insect control ability. Okay, they have insect control ability, and this is also used as commercial insecticidal agent. Now, why they are used as a commercial insecticidal agent? Because they have very low toxicity to mammals. Okay, so remember, azadirachtin is a kind of limonoid, and limonoids are triterpenes. Next, another example, phytoecdysones, which are isolated from a fern named as Polypodium. So these phytoecdysones, they are a group of plant steroids. They have the same structure as insect molting hormones. So these ferns, in response to stress or in response to pathogen attack, they synthesize phytoecdysones. So ecdysones, we all know, they are the molting hormones. Phyto means produced by plants. So when this phytoexosomes enter into insect's body, it disrupts the molting ability and other developmental processes are affected, which often lead to lethal consequences. Again, triterpenes are active against vertebrate herbivores also. We discussed till now up to insect attack, prevention from insects. But herbivores, triterpenes are active against herbivores also. So what triterpenes? Cardenolides and saponins, they are active against vertebrate herbivores. So example, these cardenolides, they are a kind of glycosides. And these cardenolides are what? They are triterpenes. Okay, they have an attached sugar, that is why they are glycosides, and they taste bitter. When they are taken in by the herbivores, okay, what happens? They cause heart arrest. What do? They have effect on the heart muscle through their inhibitory effect on sodium potassium activated ATPases, because of which they can lead to heart arrest. Okay, so remember, cardenolide inhibits Na plus K plus ATPase in herbivores. Another kind of triterpene which acts against herbivores are saponins. Okay, these saponins can form complex with sterols. So once saponin combined with sterol, they interfere with the sterol uptake from the digestive system. So sterol cannot be taken from the digestive system and it will disrupt the cell membrane structure. Okay. So few questions related to terpenes. So secondary metabolites are diverse array of organic compounds in plants. The following are certain statements about secondary metabolites. They protect the plant against being eaten by herbivores, we discussed just now, cardenolides and saponins, and against being infected by microbial pathogens, correct? Terpenes are the largest class of secondary metabolites, we discussed in the first slide only, yes. Are synthesized by MEP and shikimic acid pathway. No, shikimic acid pathway was phenol. Okay, so this is correct, but shikimic acid is wrong. So this statement became wrong. So just see how to solve the answer. So P is correct. So wherever P is not there, I'll eliminate that. So this is not my option. Q is incorrect. So Q is incorrect. That means this is wrong. This is wrong. Now let's check R. R should also be wrong, but let's check R. The most abundant classes of phenolic compounds in plants are derived from phenylalanine. Now R seems correct. Alkaloids are nitrogen containing secondary metabolites in plants. Yes, correct. So P, R and S are correct answer. Next, in terpene biosynthesis pathways. Now this question I have already discussed in the previous few slides. Okay, so please solve this on your own. Next is, following are certain statements regarding secondary metabolites found in plants. All terpenes are derived from 6 carbon element. So we discussed that terpenes are synthesized from 5 carbon element. Okay, which is IPP. So this is wrong. Alkaloids are nitrogen containing compounds. Yes. Pyrethroids are a monoterpene ester. Yes. They are synthesized in leaves and flower of chrysanthemum. Yes. They show insecticidal activity. Correct. Limonoids are group of alkaloids have anti-herbivoral activity. Now, so limonoids do not have anti-herbivoral activity. Instead, they have insecticidal property. Okay. They have